Hi, my name is Guillaume Thibault. I am a principal investigator from Nanyang Technological University, or NTU for short, in Singapore. So I want to tell you in this short video is a story that came out from my lab recently. Um, that was published in the Journal of Cell Biology on endoplasmic reticulum stress that is specifically triggered from lipid bilayer stress, or LBS. So the endoplasmic reticulum stress is a very important organelle where about 1/3 of the proteome is being synthesized, and it's also the hub of lipid biosynthesis genes as well as the storage of calcium. And when this organelle goes under stress, it activates a highly conserved stress response, which we call the unfolded protein response, or UPR. And this pathway is activated in an attempt to restore ER homeostasis. And if it goes well, the cells should survive. But then if the stress response doesn't restore homeostasis, it can lead to cell death through apoptosis. So as I just mentioned, the stress response is highly conserved from yeast to human. And in higher eukaryotes, there are 3 sensors of ER stress, while in yeast, there's only one sensor of ER stress, which is Ire1. And classically, we view ER stress as an accumulation of unfolded protein at the endoplasmic reticulum, which is then sensed by the luminal domain of Ire1. And when it sends the accumulation of unfolded protein, Ire1 will cluster together, at least from dimers, and forms higher oligomers. And it transphosphorylates itself, and then it leads to the splicing of messenger RNA HAC1 from the unspliced to the spliced version. And the spliced version will lead to the translation of the active transcription factor HAC1, and then eventually there's an upregulation of hundreds of different genes. And those genes are upregulated in an attempt to restore ER homeostasis. But then in the last decade or so, this evidence that came out from different groups that Ire1 can also sense stress independently from unfolded protein, but through the lipid bilayer of the endoplasmic reticulum, which we refers as the lipid bilayer stress, or LBS. And in that case, to the transmembrane domain, it sends the stress, and then just like unfolded protein, it leads to the same cascade downstream. But for lipid bilayer stress, most of the studies that have been done are from artificial means, either from supplementing the media with lipid precursors, such as saturated fatty acid, or by depleting a precursor of lipid bilayer components, such as inositol in yeast. So we thought that it would be very important to systematically ask what type of defect in a cell will lead to the UPR activation through lipid bilayer stress. Because in a way, we don't really understand what it really happens at the lipid bilayer that we can define as lipid bilayer stress. So we do that. To do that, we thought to do a screen, a genome-wide screen in yeast, using adapting basically a screen that has been done by Jonathan Weissman and Maya Schusdener labs, where in their case, what they have done is that they use the full-length Ire1, and then they ask for each gene that you delete from yeast, is it going to activate the UPR? And they use a GFP reporter gene for the activation of the UPR, and then it's normalized to a signal mCherry that is not modified from the screen. So to normalize the signal of the GFP to decide to score if it's a UPR activation or if there's no activation of the UPR. So we adapted the screening where we removed the luminal domain of Ire1. So in that case, without the luminal domain, which is essential to sense the accumulation of unfolded protein. So in that case, when you see UPR activation from Ire1.LD, it's only from LBS, from lipid bilayer stress. But then for full length, it can sense 2 things. It can sense unfolded protein and lipid bilayer stress. So it can sense both. So if you have a hit that is only activated from Ire1, so it means that it's from prototoxic stress. But then if it's activated from both, so it means that the common is the transmembrane domain, so it's through lipid bilayer stress. So we did that. We had two query strains, so the full length and Ire1 data LD, that we mated with our knockout library strains of about 5,000 different knockouts. And here is the result from our screen, where we identified 448 gene deletions that activated the UPR only with full-length Ire1. So these defects, I mean by the defects triggered from the lack of a gene, are activating the UPR through unfolded protein. And then in red, we found 181 gene deletions that activated the UPR for both Ire1 and Ire1 delta LD, suggesting that by removing these genes, it activates the UPR through lipid bilayer stress. But then there was also a very surprising a subset of hits that were only found with Ire1 delta LD. So we did not expect that, because we will expect that if it activates the UPR from Ire1 delta LD, it would also activate it from the full length. So there are many reasons for that can be the case. So either Ire1 delta LD is more sensitive to the defect from the deletion of these genes compared to full length. So that's why we observe a UPR activation and not in the full length. Another reason can be that from the defects here, Ire1 full length is being inhibited through the luminal domain, and you don't see this inhibition from Ire1 delta LD. And finally, it could be that somehow the luminal domain in these defects is required to restore the ER homeostasis in basal conditions through perhaps to make sure that the folding of proteins occurs. But it's not sufficient enough to see a UPR activation with the full length when you have these defects. So, but what I want to do here is to focus on the 181 genes, where the hits that were common for both, that are fully suggesting that what we observe is from lipid bilayer stress. It's probably very hard to see here from the video, but you can go back, can refer to the publication, where we have several hits with a black circle around them that were found to be hits as well from the previous screening with the full length of Ire1, which tells us that our screen is working properly in our hands as well. Then from here, what we wanted to do is to ask which type of these perturbations that we observe in yeast were also leading to DUPR activation in higher organisms, and we decided to do it in C. elegans. So what we have done in collaboration with Stephen Tarbert from the University of British Columbia, UBC, so they screen for thousands of genes where they knock down the genes and they ask if it activates UPR. And I'm just showing you some interesting hits that were common with yeast and that we followed up on them. It's genes that are required for the biosynthesis of phosphatidylcholine, or PC, which is one of the main phospholipids at the lipid bilayer. And we found 3 hits in C. elegans, PMT2, SAM1, and PCT1. And those were also found in yeast as well. And in fact, one of the highest hit is OP3 from our screen that was also found in C. elegans. So we decided to focus on OP3 as our model of lipid bilayer stress to further understand what's happening here. So for proteotoxic stress, I'm not going to show you all the data here, but we have done a lot of experiments that can be summarized here. So we use the proteotoxic stress a model where we have Ire1 knockout. We reintroduce Ire1 or Ire1 delta LD, and then we subjected them to proteotoxic stress, either by using the drug tunicamycin or DTT. And for the full length, we observe activation of the UPR, but not for Ire1 delta LD. So this activation here, which is dependent on the luminal domain, we refer to it as a proteotoxic induced UPR or UPRPT. For lipid bilayer stress, as I mentioned, we use OP3 knockout, also in Ire1 knockout as well, background, where we reintroduce the full length or Ire1 delta LD. And here we don't need to use a drug because in OP3 knockout cells, there's a constitutive a lipid bilayer stress and as well as a UPR activation. And as you can see from the summary of our data, basically either full length or Ire1 delta LD, there is an UPR activation in both because it's independent of the luminal domain of Ire1. And in that case, we refer to that as lipid bilayer stress-induced UPR or UPR-LBS. So this is kind of a nice tool to uncouple the lipid bilayer stress-induced UPR from prototoxic stress. But then what about the other way around? Can we uncouple prototoxic stress from LBS? So to do that, there were some attempts from other labs, but there was always a decrease in terms of activation overall of Ire1. So it was never as good as wild-type Ire1. So we actually found one mutation, which is very close to the transmembrane domain of Ire1. So this is the R gene just before the predicted transmembrane domain. And we think that it just sits on the surface of the lipid bilayer on the luminal side of Ire1. So this is represented with the dot, the green dot for the full-length Ire1, where we subjected this construct to this strain to tincomycin, so prototoxic stress. So there is a splicing of HAC1, which tells you about the level of activation of the UPR, so it's very strong. It's actually comparable to full length of the wild-type version of Ire1. But then on the other hand, for lipid bilayer stress with a point mutation, there's a very low splicing level of HAC1, 11%. It's actually comparable to the same construct in the presence of choline. So choline, what it does, it's a precursor of the biosynthesis of phosphatidylcholine or PC. And it does synthesize PC through the candidate pathway, and it bypasses actually OP3, so it doesn't require OP3. So OP3 is required for the biosynthesis of PC through the de novo pathway. So basically what we observe here, the splicing of HAC1 is just the same level as a background, because in the case of cells in the presence of choline, there's no lipid bilayer stress. So the level of PC is actually similar to wild-type cells, which we have done in the past. We measure that by mass spectroscopy. So that's very nice. We can couple the two. And the next question we ask is, what about the transcriptome? So is the transcriptome the same from prototoxic stress to lipid bilayer stress? And you will think that it's the same because it's a linear pathway where you have Ire1 is activated and then it leads to HAC1. And no matter what's the stress upstream, it should not matter what is downstream. But then we look at the transcriptome that taking and having evidence from literature on our previous work that it might be indeed different. And so this is just a summary from the transcriptome. Basically, we looked at which one are induced from the full length Ire1 delta LD in Ire1 knockout background, where we induce prototoxic stress using DTT. And then from our LBS side, so we had those genes that are activated from LBS. So then we were interested to look at those genes that are specifically upregulated from LBS but not during prototoxic stress. We kind of filtered away those that were common to the two. But then, of course, you can think that when you reduce the PC level in the cell, right, it will affect a lot of other things than just the UPR. So, we can have transport that is affected, other organelles, because PC is a very important phospholipid. So ideally, what you will want is to have a knockout of Ire1, so using the double knockout here without Ire1. And then you could exclude those that are upregulated independently of Ire1. But the problem is Ire1 is actually essential in OP3 knockout cells. So you need the UPR when OP3 is absent. So we could not do that. So what we did here is that we looked at the promoters of or hits of LBS-induced genes, and we found some where there was a UPR element. So the UPR element, it tells you that perhaps it's being specifically upregulated from HAC1 because this is predicted to be the binding site for HAC1 in the promoter of those genes. And those that we predicted to have a UPR element were never reported to be upregulated from the UPR. So to see if our prediction is right, so we followed up with a chip experiment where basically you look for the binding of HAC1 to those UPR elements, and you ask if it binds the promoter of the gene of interest or gene A that you see that is being upregulated. So we have done that under proteotoxic stress and lipid bilayer stress as well. And then we found 3 hits, PBI2, PR3, and PUT1, where we found that HAC1 was binding those promoters, but then not in the case of proteotoxic stress. So there was no significant binding of HAC1 for those during proteotoxic stress. In that case, we used tunicamycin. So it suggests that indeed there is a difference of how HAC1 upregulate genes either from proteotoxics or lipid bilayer stress. So there's a divergent UPR transcriptional program. But one of the criticism we got from reviewers, which was actually valid, is that perhaps what you observe here in Ire1 knockout cells is that it's acute ER stress. Because in that case, what we do usually, we incubate the cells 1 hour with either tunicamycin or a DTT. So it's a very, a strong and short response. Why in OP3? Because it's a knockout, basically, it's more like a chronic ER stress because it has been going on for a long term. And you feel like it's a bit, the UPRs are under a certain equilibrium or a steady state of the UPR activation. So it's not as dynamic as what you see here. So perhaps what you see downstream is the adaptation, while here it's not there yet. So to address this issue, what we did is that we grew the cells, OP3 knockout cells, shown in red here, in the presence of choline. So as I mentioned earlier, choline is required for the biosynthesis of PC through the Kennedy pathway. So in that case, when we grew the cells with choline, they were not under lipid bilayer stress. And then we depleted the media from choline to induce LBS in those cells where they don't make PC. And then we also, in wild-type cells in blue, we added DTT to induce a UPR. And then we followed the splicing of HAC1 over time. So with DTT is a very fast UPR activation, while with HAC1, it takes about 12 hours to see a similar splicing of HAC1. And then we ask, what about the genes that were found to be bound from a HAC1 during lipid bilayer stress, but not during proteotoxic stress? So we look at those by qPCR, and we also included Ire1, which is a very well-known gene that is upregulated from both Ire1 and OP3, and it's required during proteotoxic stress. So basically, overall, all the genes that were found from our chip experiment, they were not upregulated significantly, while Ire1 was upregulated significantly in terms of a comparison to HAC1 splicing, to see if the trend was similar, because you expect that if you have more HAC1 splice, HAC1, so you have more of the transcription factor, and then you will observe the level of the genes being upregulated as well. But then OP3 knockout cells, basically those that were following the same trend as HAC1 were the two, so PBI2 and PR3 were actually following the same trend, but not so much for PUT1. And of course, Ire1 was following the same trend. So what is telling us here, it validates our chip data that at least for these two genes, they are following the same trend, further emphasizing that Ire1, depending on the source of stress, the downstream transcriptional program that will be activated could be very different depending on the source of stress, if it's from lipid bilayer stress or proteotoxic stress. So basically what we are proposing here is that when Ire1 sends stress, either on folded protein or lipid bilayer stress, that here referred to as a membrane perturbation, then it leads to a subset of genes that some of them, they will address the unfolded protein, trying to restore homeostasis by fixing the folding problem. And other genes will be more targeting the membrane, trying to fix the membrane problem. And you can imagine that over time, one of them would be fixed and the other one might not be so much. And that what you observe is one will take over the other one. So you will see more genes upregulated from the lipid bilayer program and others from proteotoxic stress. So at the end, there's also other factors that will probably be involved that we will look at in the future, such as enhancer or inhibitor of a translation that work together with HAC1 as well. You can also imagine that HAC1 will, it upregulates some transcription factor that will upregulate other genes. So it's not just through the binding of HAC1, so there are also downstream genes as well. So if you want to know more about these stories, I invite you to scan the QR code or to go for the DOI number here to look at our publication that I oversimplified here. Or you can also have a look at our lab website if you want more information about our group and what type of work we are doing. And of course, that would not have been possible with many people involved. So this is actually a very old picture. So it's more than 1 year because, the last summer where we actually usually take those pictures, it's in the summer 2020, and we could not take a group picture because of the COVID restriction. But anyways, I want to thank the people that are in red, and especially Ian, Haoshi, and Weisheng from my lab that work on the story mostly in yeast. And also Stephen Tauber's lab, where they have done most of the work in C. elegans. And as well, Ji Hong helped a bit in some of the aspect of the work, especially for the transcriptomic analysis. And actually, this collaboration with Stephen Tauber was actually very great, and it just kind of random. So we met at a conference a few years ago, and I realized that they were doing exactly what we were starting to do in C. elegans for the screen. So since they were ahead of us, we decided to combine our effort instead of duplicating the work and competing. And I have no regrets. So again, you can just contact us if you want to have more questions or queries, or if you want more information. And I thank you for watching this video.